In statistics, the odds ratio is one of three main ways to quantify how strongly the presence or absence of property A is associated with the presence or absence of property B in a given population. If each individual in a population either does or does not have a property A and also either does or does not have a property B, where both properties are appropriately defined, then a ratio can be formed which quantitatively describes the association between the presence-absence of A, and the presence-absence of B for individuals in the population. This ratio is the odds ratio and can be computed following these steps. For a given individual that has B, compute the odds that the same individual has A. For a given individual that does not have B, compute the odds that the same individual has A. Divide the odds from step 1 by the odds from step 2 to obtain the odds ratio. The term individual in this usage does not have to refer to a human being, as a statistical population can measure any set of entities, whether living or inanimate. If the OR is greater than 1, then having A is considered to be associated with having B in the sense that the having of B raises the odds of having A. Note that this is not enough to establish that B is a contributing cause of A. It could be that the association is due to a third property, C, which is a contributing cause of both A and B. The two other major ways of quantifying association are the risk ratio and the absolute risk reduction. In clinical studies and many other settings, the parameter of greatest interest is often actually the RR, which is determined in a way that is similar to the one just described for the OR, except using probabilities instead of odds. Frequently, however, the available data only allows the computation of the OR. Notably, this is so in the case of case control studies, as explained below. On the other hand, if one of the properties is sufficiently rare, then the OR of having A given that the individual has B is a good approximation to the corresponding RR. In a more technical language, the OR is a measure of effect size, describing the strength of association or non-independence between two binary data values. It is used as a descriptive statistic, and plays an important role in logistic regression. Definition and basic properties. A motivating example, in the context of the rare disease assumption, imagine there is rare disease, afflicting, say, only 1 in many thousands of adults in a country. Imagine we suspect that being exposed to something makes it more likely to develop that disease in adulthood. The most informative thing to compute would be the risk ratio, RR. To do this in the ideal case, for all the adults in the population we would need to know whether they had the exposure to the injury as children and whether they developed the disease as adults. From this we would extract the following information: the total number of people exposed to the childhood injury, out of which developed the disease and stayed healthy. And the total number of people not exposed, out of which developed the disease and stayed healthy. Since and similarly for the NE numbers, we only have 4 independent numbers, which we can organize in a table. To avoid possible confusion, we emphasize that all these numbers refer to the entire population, and not to some sample of it. Now the risk of developing the disease given exposure is, and of developing the disease given non-exposure is the risk ratio, RR, is just the ratio of the two. Which can be rewritten as, in contrast, the odds of developing the disease given exposure is and of developing the disease given non-exposure is the odds ratio. OR, is the ratio of the two. Which can be rewritten as, we may already note that if the disease is rare, then OR per mil RR. Indeed, for a rare disease, we will have and so but then in other words, for the exposed population, the risk of developing the disease is approximately equal to the odds. Analogous reasoning shows that the risk is approximately equal to the odds for the non-exposed population as well. But then the ratio of the risks, which is RR, is approximately equal to the ratio of the odds, which is OR. Or, we could just notice that the rare disease assumption says that, and from which it follows that, in other words, that the denominators in the final expressions for the RR and the OR are approximately the same. The numerators are exactly the same, and so, again, we conclude that OR per mil RR. Returning to our hypothetical study, the problem we often face is that we may not have the data to estimate these 4 numbers. For example, we may not have the population-wide data on who did or did not have the childhood injury. Often we may overcome this problem by employing random sampling of the population, namely, if neither the disease nor the exposure to the injury are too rare in our population, then we can pick 100 people at random, and find out these four numbers in that sample. Assuming the sample is representative enough of the population, then the RR computed for this sample will be a good estimate for the RR for the whole population. However, some diseases may be so rare that, in all likelihood, even a large random sample may not contain even a single diseased individual. This would make it impossible to compute the RR. But, we may nevertheless be able to estimate the OR, provided that, unlike the disease, the exposure to the childhood injury is not too rare. Of course, because the disease is rare, this is then also our estimate for the RR. Looking at the final expression for the OR, the fraction in the numerator, we can estimate by collecting all the known cases of the disease, and seeing how many of the diseased people had the exposure, and how many did not. And the fraction in the denominator, is the odds that a healthy individual in the population was exposed to the childhood injury. Now note that this latter odds can indeed be estimated by random sampling of the population provided, as we said, that the prevalence of the exposure to the childhood injury is not too small, so that a random sample of a manageable size would be likely to contain a fair number of individuals who have had the exposure. So here the disease is very rare, but the factor thought to contribute to it is not quite so rare. Such situations are quite common in practice. Thus we can estimate the OR, and then, invoking the rare disease assumption again, we say that this is also a good approximation of the OR. Incidentally, the story just told is a paradigmatic example of a case control study. The same story could be told without ever mentioning the OR, like so, as soon as we have that and then we have that. Thus if, by random sampling, we manage to estimate then, by rare disease assumption, that will be a good estimate of which is all we need to compute the RR. However, it is standard in the literature to explicitly report the OR and then claim that the RR is approximately equal to it. Definition in terms of group-wise odds, the odds ratio is the ratio of the odds of an event occurring in one group to the odds of it occurring in another group. The term is also used to refer to sample-based estimates of this ratio. These groups might be men and women, an experimental group and a control group, or any other dichotomous classification. If the probabilities of the event in each of the groups are P1 and P2, then the odds ratio is where QXA equals A1AAAPX. An odds ratio of 1 indicates that the condition or event under study is equally likely to occur in both groups. An odds ratio greater than 1 indicates that the condition or event is more likely to occur in the first group. And an odds ratio less than 1 indicates that the condition or event is less likely to occur in the first group. The odds ratio must be non-negative if it is defined. It is undefined if P2Q1 equals 0, that is, if P2 equals 0 or Q1 equals 0. Definition in terms of joint and conditional probabilities, the odds ratio can also be defined in terms of the joint probability distribution of two binary random variables. The joint distribution of binary random variables x and y can be written where P11, P10, P01 and P00 are non-negative cell probabilities that sum to 1. The odds for y within the two subpopulations defined by x equals 1 and x equals 0 are defined in terms of the conditional probabilities given x, that is, P, yx. Thus the odds ratio is. The simple expression on the right, above, is easy to remember as the product of the probabilities of the concordant cells divided by the product of the probabilities of the discordant cells. However, note that in some applications the labeling of categories is 0 and 1 is arbitrary, so there is nothing special about concordant versus discordant values in these applications. Symmetry: if we had calculated the odds ratio based on the conditional probabilities given y, we would have gotten the same result. Other measures of effect size for binary data such as the relative risk do not have this symmetry property. Relation to statistical independence: if x and y are independent, their joint probabilities can be expressed in terms of their marginal probabilities pxa equals a p, za equals a1, and phi r equals a p, y equals a1, as follows. In this case, the odds ratio equals 1, and conversely the odds ratio can only equal 1 if the joint probabilities can be factored in this way. Thus the odds ratio equals 1 if and only if X and Y are independent. Recovering the cell probabilities from the odds ratio and marginal probabilities, the odds ratio is a function of the cell probabilities, and conversely, the cell probabilities can be recovered given knowledge of the odds ratio and the marginal probabilities P, ZA equals A1, A equals AP11 A plus AP10 and P, YI equals A1, A equals AP11 A plus AP01. If the odds ratio differs from 1, then where P1 a euro equals AP11 A plus AP10, a par euro 1 center equals AP11 A plus AP01, and in the case where I equals A1, we have independence, so P11 A equals AP1 a euro cent par euro 1 cent. Once we have P11, the other three cell probabilities can easily be recovered from the marginal probabilities. Example. Suppose that in a sample of 100 men, 90 drank wine in the previous week, while in a sample of 100 women only 20 drank wine in the same period. The odds of a man drinking wine are 90 to 10, or 9-1, while the odds of a woman drinking wine are only 20 to 80, or 1-4 equals 0.251. The odds ratio is thus 9/0.25, or 36, showing that men are much more likely to drink wine than women. The detailed calculation is. This example also shows how odds ratios are sometimes sensitive in stating relative positions. In this sample men are 90/20 equals 4.5 times more likely to have drunk wine than women, but have 36 times the odds. The logarithm of the odds ratio, the difference of the logits of the probabilities, tempers this effect, and also makes the measure symmetric with respect to the ordering of groups. For example, using natural logarithms, an odds ratio of 36/1 maps to 3.584, and an odds ratio of 1/36 maps to a 3.584. Statistical inference. Several approaches to statistical inference for odds ratios have been developed. One approach to inference uses large sample approximations to the sampling distribution of the log odds ratio. If we use the joint probability notation defined above, the population log odds ratio is. If we observe data in the form of a contingency table, then the probabilities in the joint distribution can be estimated as where pi equals an ij/an, with now equals an11 a plus an10 a plus an01 a plus an00 being the sum of all four cell counts. The sample log odds ratio is. The distribution of the log odds ratio is approximately normal with. The standard error for the log odds ratio is approximately. This is an asymptotic approximation, and will not give a meaningful result if any of the cell counts are very small. If L is the sample log odds ratio, an approximate 95% confidence interval for the population log odds ratio is L plus or minus a 1.96 SE. This can be mapped to exp, L a 1.96 SE, a exp, L plus a 1.96 SE, to obtain a 95% confidence interval for the odds ratio. If we wish to test the hypothesis that the population odds ratio equals 1, the two-sided p-value is 2p, z, and L/SE, where p denotes a probability, and z denotes a standard normal random variable. An alternative approach to inference for odds ratios looks at the distribution of the data conditionally on the marginal frequencies of x and y. An advantage of this approach is that the sampling distribution of the odds ratio can be expressed exactly. Role in logistic regression. Logistic regression is one way to generalize the odds ratio beyond two binary variables. Suppose we have a binary response variable y and a binary predictor variable x, and in addition we have other predictor variables z1, zp that may or may not be binary. If we use multiple logistic regression to regress y on x, z1, zp, then the estimated coefficient for x is related to a conditional odds ratio. Specifically, at the population level. So is an estimate of this conditional odds ratio. The interpretation of is as an estimate of the odds ratio between y and x when the values of z1, zp are held fixed. In sensitivity to the type of sampling, if the data form a population sample, Then the cell probabilities per each are interpreted as the frequencies of each of the four groups in the population as defined by their X and Y values. In many settings it is impractical to obtain a population sample, so a selected sample is used. For example, we may choose to sample units with Z equals A1 with a given probability F, regardless of their frequency in the population. In this situation, our data would follow the following joint probabilities. The odds ratio P11 P00/A P01 P10 for this distribution does not depend on the value of F. This shows that the odds ratio is invariant and non-random sampling based on one of the variables being studied. Note however that the standard error of the log odds ratio does depend on the value of F. This fact is exploited in two important situations. Suppose it is inconvenient or impractical to obtain a population sample, but it is practical to obtain a convenient sample of units with different X values, such that within the Z equals A0 and Z equals A1 subsamples the Y values are representative of the population. Suppose the marginal distribution of one variable, say X, is very skewed. For example, if we are studying the relationship between high alcohol consumption and pancreatic cancer in the general population, the incidence of pancreatic cancer would be very low, so it would require a very large population sample to get a modest number of pancreatic cancer cases. However, we could use data from hospitals to contact most or all of their pancreatic cancer patients, and then randomly sample an equal number of subjects without pancreatic cancer. In both these settings, the odds ratio can be calculated from the selected sample, without biasing the result relative to what would have been obtained for a population sample. Using quantitative research, due to the widespread use of logistic regression, the odds ratio is widely used in many fields of medical and social science research. The odds ratio is commonly used in survey research, in epidemiology, and to express the results of some clinical trials, such as in case control studies. It is often abbreviated, OR in reports. When data from multiple surveys is combined, it will often be expressed as pooled OR. Relation to relative risk, in clinical studies, as well as in some other settings, the parameter of greatest interest is often the relative risk rather than the odds ratio. The relative risk is best estimated using a population sample, but if the rare disease assumption holds, the odds ratio is a good approximation to the relative risk. The odds is par/1 AAAP, so when P moves towards 0, 1 AAAP moves towards 1, meaning that the odds approaches the risk, and the odds ratio approaches the relative risk. When the rare disease assumption does not hold, the odds ratio can overestimate the relative risk. If the absolute risk in the control group is available, conversion between the two is calculated by where RR equals relative risk, OR equals odds ratio, RC equals absolute risk in the unexposed group, given as a fraction. Confusion and exaggeration, odds ratios have often been confused with relative risk in medical literature. For non-statisticians, the odds ratio is a difficult concept to comprehend, and it gives a more impressive figure for the effect. However, most authors consider that the relative risk is readily understood. In one study, members of a national disease foundation were actually 3.5 times more likely than non-members to have heard of a common treatment for that disease but the odds ratio was 24 and the paper stated that members were more than 20-fold more likely to have heard of a unregistered trademark the treatment. A study of papers published in 2 journals reported that 26% of the articles that used an odds ratio interpreted it as a risk ratio. This may reflect the simple process of uncomprehending authors choosing the most impressive-looking and publishable figure. But its use may in some cases be deliberately deceptive. It has been suggested that the odds ratio should only be presented as a measure of effect size when the risk ratio cannot be estimated directly. Invertibility and invariance. The odds ratio has another unique property of being directly mathematically invertible whether analyzing the OR as either disease survival or disease onset incidence where the OR for survival is direct reciprocal of 1/OR for risk. This is known as the invariance of the odds ratio. In contrast, The relative risk does not possess this mathematical invertible property when studying disease survival versus onset incidence. This phenomenon of or invertibility versus R non-invertibility is best illustrated with an example. Suppose in a clinical trial, one has an adverse event risk of 4 100ths in drug group, and 2 100ths in placebo, yielding a R equals 2 and OR equals 2.04166 for drug vs placebo adverse risk. However, if analysis was inverted and adverse events were instead analyzed as event-free survival, then the drug group would have a rate of 96/100, and placebo group would have a rate of 98 100ths yielding a drug vs placebo where R equals 0.9796 for survival, but an OR equals 0.48979. As one can see, a R of 0.9796 is clearly not the reciprocal of a R of 2. In contrast, an OR of 0.48979 is indeed the direct reciprocal of an OR of 2.04166. This is again what is called the invariance of the odds ratio, and why OR for survival is not the same as OR for risk, while the OR has this symmetrical property when analyzing either survival or adverse risk. The danger to clinical interpretation for the OR comes when the adverse event rate is not rare, thereby exaggerating differences when the OR rare disease assumption is not met. On the other hand, when the disease is rare, using OR for survival can clinically hide and conceal an important doubling of adverse risk associated with a drug or exposure. Alternative estimators of the odds ratio. The sample odds ratio N11N00/AN10N01 is easy to calculate, and for moderate and large samples performs well as an estimator of the population odds ratio. When one or more of the cells in the contingency table can have a small value, the sample odds ratio can be biased and exhibit high variance. A number of alternative estimators of the odds ratio have been proposed to address this issue. One alternative estimator is the conditional maximum likelihood estimator, which conditions on the row and column margins when forming the likelihood to maximize. Another alternative estimator is the Mantel-Haenszel estimator. Numerical examples: the following four contingency tables contain observed cell counts, along with the corresponding sample odds ratio and sample log odds ratio. The following joint probability distributions contain the population cell probabilities, along with the corresponding population odds ratio and population log odds ratio. Worked example, see also, diagnostic odds ratio, forest plot, hazard ratio, rate ratio. References. External links: Odds ratio calculator, a Euro website, Odds ratio calculator with various tests, a Euro website, OpenAPI, a web-based program that calculates the odds ratio, both unmatched and pair-matched.